Hello and welcome back. We are going to keep going talking about Mendelian genetics and the stuff that isn't quite as simple as Mendel laid out. So, in the end, not everything is quite so cut and dry. But before I get into what doesn't follow Mendelian genetics, I also want to point out right here that just because something is a dominant trait doesn't actually necessarily mean it's more common. So we're going to see having extra fingers and toes is a dominant trait. It's rare, so it's not the most common. It's not the wild type trait. It's not what's most often seen, but there is no necessarily correlation between something being dominant and being inherently more prevalent within a particular population. So beyond simple dominance, we're going to learn about incomplete dominance and codominance. So we're going to learn also about polygenic, pleiotropic, and epistatic inheritance patterns. First thing is incomplete dominance. And this is another good case where you let the terms guide you. Incomplete meaning not fully. So incomplete dominance is a case where the heterozygous individual has a phenotype that's different than either of the phenotypes of the parents, meaning the heterozygous individual has a different genotype and a different phenotype that lies in between that of the parents. So here we have the offspring genotype 1 to 2 to 1. And that's what we also see for the phenotype, where you get these pink offspring from red and white. Another case of incomplete dominance we can see in humans, which is hypercholesterolemia, which is a profound disease in a homozygous recessive individual that doesn't express a particular receptor for a particular type of lipid. So that a heterozygous individual is going to have a mild disease and is intermediate between the homozygous parent. An example of codominance isn't where there's an intermediate that's between, but it's if there's two different alleles, neither is dominant and both are expressed. So they're codominant. The example that most people are familiar with for humans is blood type. So we talked a little bit before about blood types, and we talked about the fact that the difference between A and B and O have to do with these sugars on the outside of the red blood cells, where A and B are different because they have slightly different sugars on the outside. So an A sugar here you can see with the circles, and a B sugar you can see with the squares, where O has neither of those sugars. So this is codominance because we have things where you have a phenotype, and that's going to correlate with the genotype. In this case, if you have A blood, you could have both copies of A. If you had B blood, you could have both copies of B. O blood always means you have both copies of nothing, but we call this codominance because if you have AB genotype, you are going to have AB blood phenotype because both those sugars, A and B, are going to be expressed. If you have BO blood, you are going to have the B sugar on the outside along with not having that sugar. You are also going to express A blood type if you have the A allele and the O allele, which is the lack of expression. So again, this is going to be a case for blood types where it's both codominance and there are these multiple different alleles where you can have A, B, or O. That's multiple alleles, and it's codominance because if both are present, both are going to be expressed. For if you're curious for how blood typing works, this is important because we have antibodies in our blood, which hopefully now due to COVID, people are more familiar with antibodies, what they do for our immune cells. But if you have normally A sugar on your red blood cells, your body will make antibodies not to that A, but to something that's foreign to those B sugars. So if you have B type blood, you're not going to have antibodies to your B blood, you'll have antibodies to your A blood. Equally, if you're AB, you're not going to have either type of antibodies, but if you're an O individual, you are going to have antibodies to both anti-A sugars and B sugars. This is why O is the universal donor, because no one's going to be able to have antibodies that are going to react to those red blood cells, but they can only receive blood from other O individuals, because otherwise their body would respond and would form clumps with their antibodies in the red blood cells. Sometimes people ask about, well, what's the positive and negative? And that's actually a different protein. So you can read about that here. So again, you want to be able to answer these types of questions. So a man with AB blood marries a woman with O-type blood. Is there a problem? Well, if there's a problem, that's a very vague question, because in this case, of course, an O-type baby would have to have gotten an O from both of their parents. So that baby is not his child. Other complicated situations, pleiotropy. So pleiotropy is an example where you have one gene, but it's going to have multiple effects. This is something we heard about with sickle cell disease already, where there is one gene which forms one single point mutation and can form the sickle-shaped red blood cells which stick together, and has profound effects on multiple organs, causing many different symptoms, as you read or heard about from Victoria Gray in our CRISPR series. And so this is a case of pleiotropy, where one gene affects multiple different phenotypes. Another case is Marfan syndrome, which causes a mutation in a single gene, which is fibrillin, which is important for how your joints are connecting and you have loose joints. And unfortunately, there's problems with hearts. And this is one of those cases where sometimes individuals tragically die not knowing they have this condition during exercise. Polygenic traits are basically the opposite, where there's one phenotype, but many different genes are playing a role in determining that single trait. And for these types of traits that have multiple genes involved, what we typically see is what we call continuous variations. What that means is where you have a trait, so in this case, we're going to be looking at height, where in here I'm just saying 3 genes are involved. And if you take, in this case, an example, where you're looking at a P generation of very short and very tall, may have some medium height heterozygous offspring. You can see even in that F2 generation, with just looking at 3 different genes, you start to see the shape of a bell curve forming, because it's called continuous variation, because there's multiple different genes involved. Another example of this is what we see for skin color. So we all know there's a range of different colors of humans across the globe. And this is because there's more than 100 different genes products that are important for the production of color and the deposition of that color into our skin. And I love these pictures because it just shows that independent assortment of different genes. Here are 2 twin girls, so they're obviously not identical twins, but fraternal twins. So there's their 2 sisters, and here are their parents, and you can see that for skin color, it's just how that deck gets shuffled into which child got which different genes, and seeing sort of that extremes of the spectrum for light versus dark. And I will note right here that for this unit, we do have 1 extra credit assignment where you get to learn a little bit about what is the evolutionary reasons for this difference in skin colors for humans. We certainly can look at history and look at racism and seeing the effects of people's perception of what different color skin means, but we can actually learn about what the purpose of it is evolutionarily, and that's what you have an extra credit assignment on if you're interested. So another non-Mendelian genetic trait is what's actually an epistasis. So this is where it's about gene interactions in which there's one gene that is going to have an effect on multiple other genes. So we can see this in this case of coat color of dogs, but we can see it actually also with color for humans for albinism. But here we have 2 different genes. We have gene B that is going to encode melanin synthesis. And there's B that's going to produce either, if you have 2 copies of the recessive allele, that's going to be a brown dog, where if you have one copy of that dominant black allele, it's going to be a black dog. But there's this other gene that encodes a protein that is important for putting down that color. It doesn't matter if it's a black color or a brown color, but gene E is going to express a deposition protein. So what happens is if you don't have any of that active E, any big E, no color will get deposited, neither the brown nor the black, and it's those dogs which end up being the golden color. So this example in humans, there's actually the same thing, where you can have, again, a series of metabolic reactions that ultimately end up in producing that skin color, that eye color, and hair color. But in humans, there is one enzyme where if that gets mutated, all of the subsequent genes are going to be turned off. We're going to look like they're turned off. In the case of albinism, won't have any of the color deposited. So this is an example of epistasis. It's also important to point out that in addition to these things, the environment has an impact on how our genes are expressed. So there's what gets passed down in the genome, but then there's, of course, gene expression, which can be altered based on the environment, based on soil acidity, based on temperature, based on predators and signaling. And this is also true for anyone who's friends with identical twins who have the exact same genome, but they look a little bit different because that expression is different. And so that's what we call epigenetics. So the last things we need to cover are some more sort of problems in sorting things out. So we're going to now learn how to use pedigree chart to look at inheritance patterns. So a pedigree chart has these different symbols where squares are male and circles are females. And you show marriage or mating by lines across, and their offspring are coming down from them. We can form these charts of looking at how traits are passed through generations, where we're going to have the first generation, and then subsequently coming down from that, we can see the offspring. And then if those offspring mate, we can see what offspring are produced, and we can see how traits are passed down. So you can see here is widow's peak a dominant or recessive trait. You can see that widow's peak is dominant. So let's do another example. So here we can look at these different attached earlobe and free earlobe. And what we see is these free earlobed individuals, these are the dominant traits. So we only are going to get the attached in the recessive. And we can see that in this individual, if there are recessive offspring produced from a dominant parent phenotypes, that means that both those parents had to be heterozygous in order to pass down that little f. So let's do a test now. So we can see what are the genotypes for the family. We know that the attached are the shaded characters. So if the attached are the shaded, for this earlobe, we know those are all going to be the homozygous recessive. We know that they're homozygous recessive again because they had parents that had different phenotypes from them. So if you have offspring from parents with different phenotypes, that means these guys are the recessives and their parents are both heterozygous. We can continue the same way, looking at if Jill is a recessive, that means her mom, Deborah, had to be heterozygous. For both Hal and Inca, they would both be heterozygous. We know that Hal's heterozygous because he had Kevin as an offspring that has two copies of the recessive allele, so both Hal and Inna had to be heterozygous. We don't actually know the genotypes for Evelyn or Lisa, we just know they had to have at least one copy of the dominant allele. So we have our rules of the pedigree charts. You want to make sure you understand how to fill out these pedigree charts. So there's lots of different practices. We're up to the final part of this week, which is talking about genetic abnormalities and disorders. So we've talked now a lot about karyotypes. So karyotype here, we have all the chromosomes within humans here. We have autosomal chromosomes 1 through 22. We have our sex chromosomes. So we're going to be talking about different conditions for our autosomes and our sex chromosomes. So we're going to first start with autosomal dominant traits. So you're going to use the terms to guide you. If we're talking about autosomal traits, that means they're not the sex chromosomes, and it's chromosomes 1 through 22. If it's dominance, it's going to mean that heterozygous individual has the disorder. So a heterozygous parent will pass on that disorder to half of the offspring. A homozygous parent will pass on that disorder to all of their offspring. So here you can see a case where you have a heterozygous parent passing down that allele to half of their children. Some examples of autosomal dominant disorders are Marfan syndrome, which we talked about before. Huntington's disease is a particularly tragic example of autosomal dominant disorders. Another example is progeria and that polydactyly with extra fingers and toes being the dominant case. Progeria, excessive aging, and what we can see here again is that you have a phenotype ratio of a heterozygous individual mated with a homozygous recessive, so a normal individual. That phenotype ratio is going to be 2 to 2, so 50% will have the disease, 50% of the offspring wouldn't, and a genotype ratio of 0 homozygous dominant, 2 heterozygous, and 2 homozygous recessive. The next example would be autosomal recessive traits. Again, let the term lead you. Autosomal, not sex chromosomes. Recessive, meaning that only homozygous individuals are going to have the disorder. So if a parent has the disorder, that means they're going to be homozygous and they would pass on the allele to all of their offspring. And a child would only have that recessive disorder if both parents have the allele. So both parents could have the allele because they could be carriers and have just one copy of that allele, and that's called a carrier to be heterozygous. But when you do the mating for that, of course, when you mate two heterozygotes, for the genotype ratios, you get 1 to 2 to 1, and it's only this individual with the 2 copies of that recessive allele that are going to have the disorder. Some examples of recessive alleles are sickle cell anemia and cystic fibrosis and albinism. So albinism, again, is one of those cases you need to have 2 copies of that mutant allele in order not to have that protein that is going to be able to deposit the color. So you can see that example from this individual, where you have, again, a phenotype ratio of 3 to 1 and that genotype ratio of 1 homozygous dominant, 2 heterozygotes, to 1 homozygous recessive. So cystic fibrosis, I said, was an example of this recessive allele. Why cystic fibrosis is so tragic is because it is involving a mutation in a protein that's channeling chlorine outside of the cell. Cystic fibrosis is the most common fatal genetic disorder. I highly recommend clicking on this link and watching this video that's going to explain exactly cystic fibrosis. Another tragic example is Tay-Sachs disease, and this is an example where, depending on your background, you might be suggested for genetic testing because it's very common, or it's most common in Jewish people within the United States. And what Tay-Sachs is, it's the lack of a particular enzyme which results in the buildup of fats within the brain. So you can see normally fats get broken down, but when they are not able to break down, it actually kills children very young. It is very tragic. So the last cases, which are more complicated than these autosomal disorders, are X-linked disorders. So X-linked having to do with alleles on the X chromosome. And that's going to be expressed differently in males with only one copy of that X chromosome and females with their two copies of the X chromosome. And what we see is males can either have that X or that Y chromosome. They pass on the X to their daughters, they pass on their Y to their sons. Females are passing down their X chromosomes to either. Now, we mentioned this in last week's, but females have two X chromosomes. And what actually ends up happening is the body inactivates one of those two X chromosomes, forming what's called a bar body. And you can see the fact that this is sort of a random inactivation by looking at specific genes within the X chromosome and looking at different expression within an individual. You can see that there is this mosaic of some cells having one X inactivated and other cells having a different X inactivated. So what are the genes that are on the X chromosome? Well, there's actually a bunch of different genes. Some of the ones that we'll talk about involve hemophilia, Fragile X, and colorblindness. So for sex-linked recessive disorders, females with their two copies of the X chromosome, they can be normal with having just one copy of that dominant allele. They're only going to have the recessive disorder if both of their X chromosomes have that recessive allele. Whereas in males, males only have that single X chromosome, so they're only going to be normal if that contains the normal gene. If they can get one copy of the recessive allele, they will have the disease, in this case, from this graphic, hemophilia. Hemophilia is one of these super famous examples of a recessive X-linked genes, particularly from looking at the monarchy within Europe and actually the role that hemophilia played in the Russian Revolution in terms of how fragile the monarchy was. And what we see is that the females are the carriers for the gene because they had two copies of the X chromosome and did not exhibit the phenotype of hemophilia because their other X chromosome contained the normal allele, whereas the son Alexis only received that mutant allele and was thus a hemophiliac, meaning that his blood didn't clot well and he could have excessive bleeding. And hemophilia is deadly because patients can bleed out from minor cuts. So some famous X-linked recessive disorders, as just discussed, was hemophilia, but there's also some muscular dystrophies, and red blood green color blindness is also one of the most famous examples, where the more males cannot see the difference between red and green because they have the mutant X, whereas females can have that heterozygous state and just be carriers. So I've got a bunch of different problems for you to be able to work out how traits are passed down if they're sex-linked, and you can do a bunch of these problems where, again, I'll note that we write the genotypes with the X and the Y to make it clear that they're on the X chromosome, and then you just write it with that superscript of the letter. So for the last part is actually what we had already covered a little bit, but involved non-disjunction, where we talked about in meiosis, that's where the chromosomes don't line up appropriately, and you get some gametes produced that have extra copies of chromosomes. So we saw that these examples of aneuploidies, where we saw Down syndrome and some examples of the sex chromosomes being alternate versions. You might be a little bit surprised again at the frequency in which these occur. So we talked about these before, and examples of errors in crossover, where you have, again, these genetic disorders. So Fragile X syndrome is another example of this sex-linked disorders, where it's much more common in males than females. And to end, I'm not going to go through these in this video, but what I'm going to say is if you are confused on how to do these problems, click to speak to a tutor, sign up for office hours, or email me saying you want to schedule a meeting, and we can go over as many of these problems and figure out exactly what parts you are getting confused about. because I've given you tons of practice, but it's all about you actually doing it. So I wish you well. I hope to hear from you if you are confused. Reach out and use the resources provided. Be well.